Good day, Homo sapiens. Welcome back to my channel. And today we are studying once again, the microbiology book. And today's topic is preparation and staining of specimens. And we are going to study the different staining techniques like, differential staining, special stain, and simple staining, specifically gram staining, okay? And we are not just going to study types of stains, but we are also going to study the dyes. You can say stain or dyes, right? And you are going to study the techniques as well, like how do we use them actually, and the other thing is how do you prepare your specimen. That's important as well, and not to be taken lightly, because mostly the error there causes error in the staining as well. So, well, I think most likely we will be covering that today. So based upon, and these are not that difficult, and so you are going to get covered with these, because these are very commonly used in microbiology, and specifically when you do microscopy. So without any waiting, let's start. But wait, oh sorry, I need to check if this is working or not. Wait, just give me a second. I need to check with these, because these are very right. It's working. So I'll just here, and let's go back. Alright, let's start. If you have any question, you can ask me about staining. I'll cover it. So, preparation and staining of specimen. By the way, I'm reading the Prescott Microbiology if anyone is intrigued or wanting to know about this. Although living microorganisms can be directly examined with the light microscope, they often must be fixed and stained to increase the visibility, accentuate specific morphological features, and preserve them for future study. Alright guys, so they are saying if you want to study the living microorganisms, microorganisms under the light microscope, so you first need to fix it, you cover it, or like if the specimen shouldn't move here and there, it should be fixed, and you should stain it, because the visibility of microorganism is very light due to their light coloration. Specifically, some microorganisms don't even have color that much stain, and they are mostly invisible. So not just increasing visibility, but also making it more prone to showing some features more prominently and some less prominently, so you know what type of organelle you stained, and so your focus is only on that organelle and not on the other organelles. So that's why we are going to study different types of stains, and we all will know that if we use this stain, it will stain these kind of features. Morphological means the structural features. We will be able to see this. If you want to study the whole structure, there is a different types of stain for that as well, and preserve them for future study. I didn't know that staining could be used for preservation. Maybe they are talking about different. We are studying it further. If we don't, we will search it. So fixation. First process is fixation. Before staining, you have to fix the specimen, right? The specimen that you want to study. The stained cells seen in the microscope should resemble, give me a second, should resemble living cells as closely as possible, right? First of all, if you want the stained cells that you colored, want to see them looking as realistic, as clear as they were as a living cells, the fixing is the first thing to do, right? So the fixing process make it possible for them to resemble the living cells as closely as possible. Fixation is the process by which the internal and external structures of the cells and microorganisms are preserved and fixed in a position. Exactly. It preserves it and fixes it. But the thing that they added is the internal structures and the external structures. Both these structures, I mean the organelles, or you can say the cell membrane, cell walls, or anything that is, you know, it is still in a freezing point. I'm freezing, talking about freezing because I watched movies a lot about the time stopping or things. So it inactivates the enzyme that might disrupt the cellular morphology and toughens the cellular structure so that do not change during the staining and observation. By the way, they remember that staining is quite rough on some kind of cells. So the fixation process actually make it possible for the cells to adhere to the surface, or you can say, and to and in strengthen the cell to be able to cope up with these highly reactive, or you can say strong acids or dyes. It can be basic though. So alright, and so the other thing that fixation does is inactivates the enzymes, of course. Unless the cell is dead, the enzyme won't stop working, right? But here we are studying the living cells, and the living cells do have enzymes that work 24/7. So it inactivates all of these enzymes that might disrupt the cellular morphology, right? So there are some enzymes that may be making the cell membrane, and so if it continuously work, the cell membrane will be continuously changing. As a result, will make it difficult to study under the microscope. So things like that, right? Enzymes like that. A microorganism usually is killed and attached firmly to microscope slide during the fixation. Alright. So they are saying usually the microorganism is killed. Like this is the easiest way for the microorganisms to get fixed, because if you kill them, automatically their enzymes will stop working. Automatically their cell morphology won't change, right? So they usually are killed, but there are some cases where we need to study the living cells, right? How the cells organelles actually work or things like that, so we don't kill them. And then we attach firmly to the microscope slide. Yeah, it needs to be firmly attached. We don't want it moving here and there and making the clarity of the slide bad. So during fixation, what two things we do? We either kill it, or either put some kind of material and it inactivates their enzyme and make the cell into a suspended state where it looks like that, but it is living organism. And the second is attachment. You have to attach them to the slide. There are two fundamentally different types of fixation. Alright, now they are telling us heat fixation is routinely used to observe prokaryotes. So by the name of heat, it is, I think, quite visible that heat is for killing most of the microorganisms. And by prokaryotes, this means bacteria, right? Or archaea. So typically a film of cells, a smear, is gently heated as the slide is passed through the flame. Alright, so in heat fixation it is passed through, the heat shouldn't be that strong, or so it doesn't harm the cell's internal structure as much, so it should be passed gently through the flame. Heat fixation preserves the overall morphology but not the structures within the cells. Excuse me? What does this means? Heat fixation preserves the overall morphology, right? The overall morphology, but not the structures within the cell. Oh, right, I get it. Like it can harm the cells inside the cells. For example, there must be proteins like enzymes. They are very temperature sensitive, and it does, get, even if you gently pass it through the flame, it will get damaged. So wait, let me still search it. Like, what does this sentence mean? I wonder if it will be answered. Heat denatures the proteolytic enzymes and prevents the autolysis. Heat fixation cannot be used in the capsular stain method, as heat fixation will shrink or destroys the capsule and cannot be seen in stain. Heat fixation, or about nothing. means that while using heat to fix a biological sample, the general shape and outline of the cell will be maintained, but the internal components and detailed structures will be damaged due to the high temperatures, making them difficult to observe under microscope. Right. I like it's the normal thing. The internal structure are sensitive to heat, unlike the outer membrane cell walls. I mean, they are more resistant or more reluctant to damage than the internal structures. So the pro is it is easier and simpler to adhere the cells to the slide, particularly useful for bacteria. That's why they use it is commonly used for prokaryotes, right? By essentially cooking them on the surface. And cons, yeah, because of heat, it can disrupt the delicate internal structures like organelles, making them unable to. Right. Cellular analysis is not, for the heat fixation. The second method is chemical fixation. It is used to protect the fine cellular substructure and the morphology of larger, more delicate organisms. Alright, so this is advanced method than the heat fixation, but it is more complex and use chemicals. So it fixes the overall morphology and the inner substructure, right? Or you can say substructure as well. And chemical fixatives penetrate the cells and react with the cellular components, usually proteins and lipids, to render them inactive, insoluble, and immobile. Alright, so I think these chemical fixatives are actually the chemicals that we talked about to suspend the cells in a freezing state where their enzymes becomes inactive. And I think this is done for us if we want to study the cell in a living condition. That's how we will be able to study their inner cellular components and how they actually work. So they attach to the proteins and lipids. Like in case of proteins, don't think like proteins are just simple components. Enzymes, think about enzymes become inactive. Lipids are the carriers, transporters, and they become inactive as well. Like most work done by these two type of things. Most like we do have minerals and these kind of things, but if they don't work, they stop. Like their working isn't that dangerous, or you can say it doesn't drastically change the cell. Inactive, insoluble, and immobile. Right. Common fixative mixtures contain such components such as ethanols, acetic acid, mercuric chloride, formaldehyde, and glutaraldehyde. Well, so this is the example that fixative mixtures have this ethanol, acetic acid, mercuric chloride, formaldehyde, and glutaraldehyde. Although I don't know them personally, I think at the end of this live, I will search them in the Google, right? I'm curious what these are, like, what these things are, what these chemicals actually are. Even though I know we use it in a chemical fixation. So, dyes and simple staining. Now here comes the staining part. That was just the fixation part. Making the cell attached and immovable, inactive. That was the fixation part. Here is the visibility parts coming, right? And for that we use dyes. And first comes the simple staining. Like just you don't want to differentiate them, you don't want to classify them, you just want to see the structure, right? That is, I think, simple staining. So, the many types of dyes used to stain the microorganisms have two features in common. Alright. So, the many types of dyes we use have two things in common. They have chromophore groups with conjugated double bonds that give the dye its color, and they can bind with the cells by ionic covalent and hydrophobic bonding. So they have chromophore group. By the word chromo, you will be getting that chromo means colorful, just as chloro means green. Chromo means colorful, can be any color. So these are the chromophore groups. Chromophore groups. These are the dyes, right? By name it gives the color. And how does it give color? By making the ionic bond or covalent bond or hydrophobic bond, right? And how is their internal structure? The groups with conjugated double bonds. Guys, this is not the conjugate for This is not the conjugate for, a simple English. This is the chemical term. Conjugates the complex that is formed. And I think they form that kind of conjugate double bonds. And that's how they gain its color. Conjugated double bonds. Wait, let's search it on a Google. It's better that way. Are double bonds that alternate with a single bond in an organic structure. What? Excuse me? Was it that simple? Give me a second. I didn't know it was that simple. Oh, I was thinking of molecular structure. And that might have been not simple. Was I thinking something else? It's as simple as that. Double bonds that alternate with a single bond, like one double bond, one single bond, one double bond, one single bond. But why does bonds gives chromophore characteristics? Whatever. Let's not look at the spellings. Indicates a region in the molecule where the energy differences between two separate molecular orbitals fall within the range of visible light. Oh. As conjugation increases, the molecular orbital energy decreases so that the phi electron transition occur in the UV and visible region of the. Alright, so the energy levels, we all know that the energy is released in wavelengths, right? There's a pocket of energy and, it's like it has its own wavelength, and there is certain time that it occurs in the visible region of light, right? Yeah. I think you kind of get it. That these are, chromophore groups are the groups in the molecules. The first that has conjugate double bonds, mean one double bond, one single bond, one double bond, one single bond, like these kind of pattern. And then the other thing is their inner electron structure is like, transition like that it reflects only the visible light. So by it reflects the visible light in certain way that makes it colorful, just as the leaves reflects the green color, so that's why the leaves looks green, and that's why it's a chlorophyll, right? And in chromophore it might reflect red color or things like that depending on the dye that we use. Alright, so ionic bond, covalent bond, hydrophobic. By the way, these are quite strong bonds, and ionic, covalent, are hydrophobic, because this is not van der Waals force, or what was the other? Polar? That is covalent bond. There was another one. London dispersion, yeah, these are the weak forces, these are the strong forces. So most dyes are used to directly stain the cell or object of interest, but some dyes, they are saying some are used to direct, stain. Like once you put it, these cells are, stained. But some dyes, for example, India ink and nigrosine, are used in negative staining. Guys, negative staining is that once you put a stain on certain cell, it doesn't retain that stain after you wash it, right? That's why you need one more stain for that, and that becomes double stain, right? So that's why we call it negative staining. Like the first stain is not retained, that's why we need another stain, and that's why it makes it negative staining. Like it doesn't go on the first step. If it goes on the first step, it's positive staining, like we are certain that it is stained. But when it doesn't, it becomes negative staining, and we need another stain for that. This is my logic for remembering it. Like, there is positive staining and there's a negative staining. So there are dyes that we do use for gram staining, right? We use these terms in gram staining. It will come more forward. So they are saying some dyes are used directly on the cells or objects of interest, and it does retain its stain. But some dyes are used in negative staining, where the background but not the cell is stained. The unstained cells appear as bright objects. What the heck? Oh my god, this is embarrassing. What the heck was I doing? Can we delete what I said just now? But this is live, it can't be deleted. Why do I not read things like that? The negative staining wasn't what I was saying. It was a different story. Forget that. Completely remove it from your mind, alright? Negative staining is when the background is stained but the cells are not stained, so it is indirectly making the cells visible because the background is just so so dark that the invisible things appear more bright. Alright, so this is the correct explanation. Forget about the other one. Dyes that bind the cells by ionic interactions are probably the most commonly used dyes. Alright, so they are saying the one that uses the ionic bonding are most commonly used dyes, and why? These ionizable dyes may be divided into two general classes based on the nature of their charge. Yeah, I have heard of these basic dyes and acidic dyes. They definitely are used commonly. And yeah, the naming is as shown, the basic and the acidic, because of their ionic differences. So methylene blue, basic fuchsine, crystal violet, safranine. Alright, I know the safranine, which is used in gram staining. Malachite green have positively charged group, usually some form of the pentavalent nitrogen. So they are saying that they are positively charged. So basics are positively charged, acidics are negatively charged, and they have pentavalent nitrogen. Nitrogen usually makes 7 bonds, right? What does pentavalent mean again? You guys must be thinking what the dumb channel I went into, but it is, study together, so don't think I'm your teacher or something. Typically only available to form 3 bonds. Yeah, that's what I was saying. The nitrogen makes only have total of 7 electrons. Originally it does have 7 electrons. No, its valence is 7. That means it makes outermost 1 2 and then 1 2 3 4 5. It does makes 5 bonding. Guys, skip this, okay, if you want to study about just stainings. I'm curious, what do they mean by this staining? Pentavalent. Valence, the number of chemical bonds. Nitrogen valence is usually 3. What the heck? 2, 2, or 3. Alright. S has 2 electrons, and the 1s has 2 electrons, 2s has 2 electrons, and so of course P has only 3 electrons. That's why it makes only 3 bonds. But no vacant d orbitals. Alright. But what happens to the pentavalent nitrogen? It makes 5 bonds. Alright, so all of them, the S goes into the S1 nitrogen. How do I show it to you? I finally get it. Here. Guys, let me explain it this way, alright? So this is nitrogen. Wait, let me change the color so we can see it. So this is our nitrogen, and it usually have s orbital, 2s orbitals, and p orbital, right? Like you can say, so 1s has 2 electrons, 2s has 2 electrons, and 3s has, 3s not 3s, 2p, and p have px, py, and pz. Py and pz, and all of them has, you know, 1 electron. Like the 2p shall have total of 3 electrons. So in total 6, 5, 6, 7. The nitrogen has total of 7 electrons, right? That's the nitrogen. And after nitrogen, oxygen comes and it has total of 8 electrons. Right. So this is how the nitrogen makes 3 bonds with another nitrogen, right? This is usually how it happens because this is satisfied and this S doesn't make any bonding. But in not normal cases, this S, P, X, P, Y, and PZ, right? So the 2 electrons that was in the S, the one that was in the S goes to the d electron, d orbital, right? It goes to the d orbital. So the nitrogen makes total of 5 bondings. 1, 2, 3, 4, 5. With another nitrogen, or maybe with another, you know, well, it's difficult for it to attain another nitrogen. So let's forget this was nitrogen. Consider this as H and some other elements, or you can say atoms. So it forms bonding with another. So there is total of 5 atoms in this, right? So this goes to the D, and so total of 5 bondings is formed, and this makes it pentavalent, right? Like it has 5 bondings in the valence shell. While in usual cases the S is satisfied and it has 2 electrons and it doesn't make bonding with another one, and so the total of bonding in the valence shell is 3. So like it is usually making 3 bonds, but in some cases, rare cases, it makes the pentabonding. I forgot what was the reasoning for it to make, for it to go from the, you know, the, what do you call it, 3-valent to pentavalent. What was the reason for it to go? But they are saying that these have positively charged group and usually some form of pentavalent nitrogen. Right, so they are saying in these kind of dyes there are cases where there is pentavalent nitrogen involved for the positive group, and making it basic dyes are generally sold as chloride salts. Chloride salts. Why chloride salts? Oh, chloride salts. Basic dyes bind to negatively charged molecules like nucleic acid, many proteins, and the surface of prokaryotic cells. Alright, so this part we do get it, that the basic dyes binds to negatively charged molecules. Nucleic acid is negatively charged because of its P phosphorus, right? Get it? P nitrogen. What am I drawing? How does DNA looks again? P sugar. Yeah, sugar and nitrogenous base. Again P, like one is attached to this and one is attached to this one, and then another sugar is attached to this one and another sugar is attached to this one, right? Right, consider this a 5 sugar. I know I'm drawing it a little bit roughly. This is the original how the ribose sugar looks like. So this is how it looks like, and these P have negative charge on them, and so that's why the nucleic acid have a P negative charge on it. So that's why the positive basic dye is attached to the nucleic acid, while proteins, where are proteins? Yeah, proteins, many proteins. Yeah, there are proteins, you know. Some amino acids are negative, some amino acids are positive. So yeah, it attaches to many proteins, and it's a and because we have the proteins in the lipid bilayer of, you can say the, what do you call it, the outer layer of the prokaryotic cells. Wait, what in the outer layer of prokaryotic cells are negative? What is negative in prokaryotic? I wrote the spelling wrong. Wait, let me think. Oh, no. cell membranes. No, I wasn't talking about this. I'm saying, what is the negative material? Ah, okay. Oh, alright. The lipopolysaccharides are actually negative. Oh, yeah. It does have phosphate groups, don't they? Wait, they did give explanation for it. Giving them, which are the major giving negative. The outer membrane is considered the negative material due to its distinct composition composed to the inner cell membrane. Wait. Why is LPS negative? Because it contains multiple phosphate groups. Yeah, I was right. It was because of phosphate groups, particularly in lipid A company, which contribute. Okay, whatever. We are not going that much deeper into the structure, but it is because of the phosphate groups, and it was because of the phosphate group in the the nucleic acid as well. All right. So what I wanted to search was the chloride salts. Definitely the chloride salts. I want to look, or I want to see what the chloride salts are look like. Sodium chloride, calcium chloride, chloride salt as stains. They did show me. Why did I change the image? So this doesn't look like it is used as, colors. This one will looks nice, right? Let me enlarge it a little bit. Hey, you go. So these are the types of dyes I think they use. So cobalt, the first one is, you can see the brown one, the cobalt chloride. The second is the copper chloride. The third is copper sulfate. The greenish one is ferrous sulfate. The fifth one is ferric chloride. It is brown again, blackish brown. And the potassium dichloride is the orange. Wow, such a beautiful color. And the nickel chloride. Guys, I don't know what's wrong with my throat. Alright, so we are good with it. So these are chloride salts, and I don't know methylene blue. Wait, let me search for their whole names. Is the names that did the names that we just studied, like these names, the names matches with these names? Oh, no. Let me real names. Alright, so these are the real names. Guys, tell me the real names. I just want to know if the names that we mentioned does match with this or not. Do I have to... Let's see what is the name of methyl blue. Or common name. Chloride. Yes, it is the chloride. It is the chloride. Alright, so let's go with the basic fuchsin. Control plus C and control plus V. Hydrochloride. Look at this red color. Alright. The next one is crystal violet. Although we know what this is by the control plus C, I'm still going to search what the real name is. It looks like all of them are chloride. Oh, what is this? Dimethyl. It isn't chloride. The name chloride isn't in it. It is a toxic. Alright, so crystal violet is what? I forget my spelling again. Hexamethyl pararosanilene chloride. So it is chloride. I didn't know its full name was this. And the last one is safranin. It is chloride as well. Dimethyl 5-phenyl phenazinium. It is also known as basic red 2 safranin O, safranin T. So many names for just one color. Alright, so these are the basic dyes, and they bind to the negatively. Since they are positive, they bind to the negatively charged molecules like nucleic acid and proteins and the surfaces of the prokaryotic cells, while the acidic dyes, eosin, rose bengal, and the acid fuchsin possesses negatively charged groups such as carboxyl and the phenolic hydroxyls. Alright, we did said that there were the positively charged groups, and usually some form had the pentavalent nitrogen, while in this case some has the carboxyls and the phenolic hydroxyls, right? Hydroxyls. So looks like the oxyls group, OH group, is more dominant or more prominent in the acidic dyes. Acidic dyes, because of their negative charges, bind to the positively charged cell structure. We get it. We are not going to search the eosin, rose bengal, and the acid fuchsin. The staining effectiveness of the ionizable dyes may be altered by pH, since the nature and degree of the charge on the cell component change with the pH. That's correct. They are saying that the effectiveness of the staining will change if we change the pH, because pH are hydrogen plus ions, right? Is the concentration of hydrogen plus ions and the OH plus ions, mostly that concentration it measures. So if we change the pH, the ionizing, ionizable dyes. Should I stop here? Looks like I have to stop it here. I can't continue. By the way, I'm completely fine. It just happened, and I think I need to take a break. So we will study the remaining on the next day. So by the way, we studied this methylene blue stain, right? Methylene blue stain, and this is stain on the corine bacterium. It's a bacterium. And look at this crystal violet stain on Escherichia coli. Escherichia coli we all know, look at this. So beautiful color. And what other stains did we study? Alright, so the others are differential stains and the special stains. And the only two types of stains we studied till now is A and B, crystal violet and the methylene blue stain. Right, so these are the simple stains that we studied till now. Right, so this process that is shown here is the process of the gram staining, and we will study it the next day. Oh wait, I am fine now, I can continue. If it starts again, I'm stopping there. Since the nature and degree of the no, I am not fine. Alright, good day, that was it, and yeah, I'm ending the stream. And if you wanna see the part 2, you can, search it. After 2 days, it will be out.